The overall goal of this Conductimetric Biosensor Assay is to detect biomolecular interactions using the hybrid β-lactamase technology.
We describe a method to detect antibody-antigen interactions using a chimeric beta-lactamase protein. The principle of this method relies on detecting binding by measuring the proton release of the beta-lactamase activity. The main advantage of this technique over conventional immunosensors is that the use of a chimeric beta-lactamase makes cheaper and faster sensor assays by using a hybrid protein to detect the analyte. Demonstrating the procedure will be Mathieu Dondelinger, a graduate researcher.
Begin this procedure with protein sample and solution preparation as described in the text protocol. Prepare the reusable electrode by dipping the tips into wells of a 96-well plate containing 300 microliters per well of electrode preparation solution for 2 minutes with gentle mixing at room temperature. Repeat this step 3 times. Then rinse the electrodes by dipping the tips into wells of a 96-well plate containing 300 microliters per well of distilled water for 2 minutes with gentle mixing at room temperature. To regenerate the electrodes, dip the tips into wells of a 96-well plate containing 300 microliters per well of regeneration solution overnight at 4 degrees Celsius or 1 hour at room temperature. Then perform 3 washes of the electrodes by dipping the tips into wells of a 96-well plate containing 300 microliters per well of phosphate-buffered saline. Perform each wash for 2 minutes with gentle mixing at room temperature.